cell is calculated and the length of the DNA calculated simply by multiplying the total number of base pairs, distance between the two consecutive base pairs. So in the mammalian cell, 6.6 into 10 to the power of 9 number of base pairs are present. Apart from this, into 0.34 into 10 to the power of minus 9 millionth of a base pair. Okay, it comes out to be approximately 2.2 meters. How much it is? 2.2 meters. Okay, so let us imagine this much of DNA base pair content is there. Okay, if you take out all the DNA content from our body, we can wrap around the sun 600 times. That much DNA is there in our cells. Approximately 70 trillion cells are present in our body. A length, if you take the length, that is far greater than the dimension of a typical nucleus. It is far greater than the length of the DNA is typically greater than the nucleus. Okay, approximately 10 to the power of minus 6. Okay, now in a prokaryotic cell such as, let us take the E. coli bacteria. In a E. coli bacteria, they do not have any defined nucleus, isn't it? Nuclear membrane is absent, hence genetic material is dispersed in the cytoplasm. The DNA, if you carefully see, is not scattered throughout the cell. So, DNA is a being negatively charged because of phosphoric acid is held with some proteins have a positive charge in the region termed as a nucleoid. Okay? Negatively charged DNA wrapped around the positively charged proteins. DNA in nucleoid is organized in large loops by proteins. Here is the central dogma proposed by Francis Crick. DNA through the transcription it produces mRNA. mRNA through translation it produces the protein, which is considered as a central dogma for the genetic material. And in eukaryotes, this generation is much more complex. Okay, there is a set of positively charged basic proteins called histones are present. Histone proteins are present. A protein acquires charge depends upon the number of and type of amino acids present in it. Okay, now resulted in the charged side chains. Histones are rich in the basic amino acid, and if you take the lysine and arginine, if you carefully see, these both amino acids residues, they carry positive charges in their side chains. Histones are organized to form. If you carefully take the histone proteins, histones are organized to form a unit of 8 molecules called histone octamer. Okay, what is that called? Histone octamer. Okay, so this histone octamer, if you carefully see, later on I will explain the histone octamer later on. The negatively charged DNA is wrapped around the positively charged histone proteins, okay, to form a structure called nucleosome. Apart from this, a typical nucleosome contains 200 base pairs, okay, of the DNA. Nucleosomes constitute repeating units of a structure in nucleus called chromatin, and thread-like strands are present here. Colored body is seen in the nucleus. The nucleosome, if you carefully see the nucleosomes in chromatin are seen just like a beds on string structure when viewed under electronic microscope. The beds on a string structure in chromatin is generally packed, okay, to form a specific chromatin fibers, okay, that are further coiled together and considered at a metaphase okay, at a metaphase cell division during the chromosome appearance. This package, if you carefully see, this package of chromatin, if you carefully observe it, at a higher level requires a set of proteins, okay. These set of proteins, okay, that collectively referred as non-histone chromosomal proteins. Here, in a typical nucleus, some regions of the chromatin matter are loosely packed, so they are loosely packed and are referred as a euchromatin In this case, the chromatin that is more densely packed and Stein's dogs are called as a heterochromatin. Euchromatin is said to be transcriptionally active, which is nothing but it is an active chromatin, whereas heterochromatin is inactive transcriptionally. What is transcription? I will tell you in the coming videos. Okay? Until stay tuned, don't forget to subscribe.